Okay, you rejoin us aboard the RV Falcor. The time is just going up to 9:30. We are at a depth of 4,998 meters. We are heading due south, and we can see what look suspiciously like pillow basalts. This is good news. We know that immediately above the contact, as we come up this slope, we will see we have pillow basalts here. The lobate structure there, you can see it's a much smoother surface than we've seen on the angular hydrothermal setting of the Von Dam mound, and also of the sulfide mound that's at the top of this hill, which underpins the geologic setting. This is the world's deepest hydrothermal field, lies just uphill from here, but it sits on top of a large volcanic mountain, so the contours drop away beneath us and to the right, and steeply downhill. It goes down about another 500 meters. So we're climbing this tall volcanic mountain now as we continue south, and as we get to the top of the volcanic mountain is where we find the hot springs. And in particular, where we're headed this morning is to the northern flank of the BB Vents mound. The top of that mound, a tall sulfide structure, is bristling with 5 high-temperature chimneys, slender chimneys, each one of 5 to 10 meters tall, spewing fluids that are approaching 400 degrees Celsius. But all over this north flank of the same mound, there's extensive hydrothermal activity, which is slightly lower temperatures. So I think this is the deepest point we've been to with Nereus so far. We're at exactly 5,000 meters right now, and ascending the hillside. And you can see the view is turning immediately from, it was all pillow lavas, you can now see angular fragments cascading down the hillside. More sediment covering as we move up. And these large angular blocks, we believe, are predominantly metal sulfide ore, which has been shed from the top of the mountain where the active venting occurs. What we're on the lookout for now, both in the field of view that you have on the internet feed and also on our other peripheral cameras, is evidence for diffuse fluid flow. So we're looking for, the easy way to find that is when you see light patches consistent with biological activity against a dark and rather dull background. So moving up slowly over these lobate sort of blobby things you're looking at here are definitely more pillow lavas. I see diffuse flow directly ahead in the far field camera. It doesn't have any markers. That's probably the first of the diffuse flow sites I was hoping we were going to find today. Kind of just like we planned. So let's settle up and have a look at that. We don't necessarily have to do anything right now, but I think if we stay long enough and drop a DVL target for this, we can always go out back to this. Actually, is this actually... No, sorry. I was caught by the bright mineral. Okay, sorry about that, Casey. A false alarm. I saw something light, but it's actually the mineral texture of a recently cracked open lump of either rock or hydrothermal deposit. You can see the difference now. This large block that's we're going to be scouting around the side of Has a very ribbed texture. That's very clearly not basaltic material. That's definitely sulfide chimney material. You see these huge amounts of this stuff all over the seafloor here. So we're scouting our way carefully and cautiously up and across this to the left. We think the hot chimlet site is probably due west of us, or due east of us now. Looks like a pair of shrimp on that sulfide chimney, so although the chimney's clearly dead sulfide, if there are two of our Rimicaris shrimp nearby, that's a reasonably promising sign that we're not too far off target. Now just lifting back away from the edge, you can see the hillside isn't a completely flat and smooth hillside, it's a series of ravines and gullies. So just stepping away from one promontory, getting back a healthy distance out into the water column so we can actually look at the next section across as we get ourselves closer to where we ultimately want to be. Fish here on the seafloor. Our water depth now is 49.90, which was our target depth. And we're within 5 to 10 meters of where we'd seen our hot chimlet site previously. And I think, is that a marker up there on the left? So we think in the, when we panned up briefly there, we could see a large white object in the distance, which we're pretty sure is our own man-made marker. There isn't much that's that bright and white around on the seafloor, unless you are right at certain kinds of hydrothermal field. So we're pretty happy that we're coming to the right spot. What you can see dropping away underneath the basket right now, though, shows you the difficult terrain that we're actually working in. It's almost a vertical cliff dropping away here. That's why we made sure we didn't go all the way to the seafloor and climb the entire hillside. We actually hovered off out in mid-water and then proceeded slowly and cautiously in towards the vents. Okay, very good. We think now, despite a couple of false alarms, we think we really do have something interesting to show you at last. And in fact, it's another false alarm.
We're getting showered.
I think 4988 is... We see a glass sponge attached to these rocks, the large overhangs of sulfides. There's something around the far side of the rocks that hopefully isn't just more false alarm white minerals. You can see these giant overhanging rocks can be quite confusing. It's all very well to know you're within 5 meters of a vent site, but when there's a 10 meter tall, 10 meter large rock right in your way. So this will be that, with that marker line, that'll be the whalebone. So what I'd like us to do, what we do know is that the hot chimlet site itself is just beyond that, kind of over to the left in the same orientation. Yeah, but I think, yeah, what I'd like us to do is not go too fast as we approach this, is just check out and see if there are any other diffuse flow sites between here and this marker, and then and if not, we'll carry on to the other marker, and that's where we're going to put out our Josh Simpson planet on the seafloor as part of the Infinity art project. So we're arriving in the hot chimlets area. When we were here in 2012, what we actually noted was that there were 3 sets of diffuse flow, and it was the uppermost of the 3 that we sampled, and that's where we put out our marker. What we're just checking now is we know that the second marker that's been put in place is from Adrian Glover and Diva Amon from the Natural History Museum in London. At the bottom of their anchor there is actually a set of whale bones, but just in the foreground here is a site of diffuse flow. I'm just going to have a quick conversation with Casey. Okay, James, our navigation guru, is confident that he's brought us to the right spot. He trusts us not to be able to mess things up from here, so we're going to let him go sleep for both hours, and then we'll get him up later when we're doing more complex maneuvers again later on. In the meantime, we are just setting ourselves up. Casey's first impossible task of the morning, apart from the fact that we landed him right smack bang in a cliff face, is going to be to perch and hover and try and measure the temperature of the fluids in and around a little patch of shrimp we've seen, which themselves are just downhill from where the whalebone experiment has been deployed by our colleagues from the Natural History Museum in London. So as the dust begins to clear, we can see what we've come across here is a site of with shrimp hanging around on porous sulfides. In amongst the dust we can also see there's a lot of shimmering water with the fluid flow leaking out of the hillside. You can see little white rills of patterns around across the seafloor, basically the plates of sulfide with fluid coming out from underneath them. So as the dust cleared in the distance, we realised there's actually a stronger patch of flow, or certainly a higher concentration of shrimp. Actually, what's that down the bottom left there?
There is float in there.
Is that within your reaching distance right now?
Should be.
Or at least can we switch to the zoom camera and have a look at that? And it probably won't do any harm to get the temperature wand in our hand, because we can probe that one and then we can move up the hill to the other. Does that sound like a good plan? I can see some bright white mineralization inside that hole. So I think what I'd like us to do would be to take a temperature measurement in that hole, please, and see what's there. Yep, this is Nereus Dive 62, sample 1, we had temperature recording. And while Casey sets up for that, I'm going to go. The temperature reading will be the first sample. Sorry, I just wanted to make sure there was active flow. We were just zooming in there rather than for the sake of photogenics. So here we are aboard the RV Falcor with the OV Nereus, about to make a temperature measurement at a site of fluid flow at the very deepest point of the world's deepest hydrothermal field. And in fact, we're slightly downhill from the hot chinlet site, so this is the deepest of the deep. 85, did you say? 25. I get the impression you probably want to rotate that way and go that way. Okay. I don't think 21 degrees is going to be hot enough to get anyone too excited today. Jeff, you're looking for 80 degrees as your ideal sweet spot for today, right? No. Would you mind going across to the library and asking her? It's certainly a very stable flow that we should be able to study. So we have a T-max of 22 degrees so far. We have another site just upstream from here, but if this is of interest to her, Casey, then this is actually the right inlet. We could just start the sample straight away. How's that for ruthless efficiency?
We're ruthless.
We've got 5 hours on the seafloor and 30 minutes in. So we're in very good shape. So what you can see living around this fluid flow is warm water shimmering out from the hillside. The fluids, we know the end member fluids that produce the black smoke are very rich in minerals and hydrogen sulfide and metals. By the time it comes out in this form, if it's just clear water shimmering at these temperatures, what we know is that a lot of the metals will already have precipitated, and that probably explains how it is that we have this large metal mound. Basically the orange color you see is the iron rusting away from the hydrothermal field. So this is not super great for measuring high metal concentrations and looking at geochemistry from that perspective, but they're very interesting in terms of microbial activity and in terms of shrimp. So Jeff went off to ask Julie if she was interested, if the temperature was around 25, and she said no, but if it was warmer it would be good. Jeff came back and his temperature probe showed the temperature had gone up to 35, so he's gone back to ask Julie a second time. And maybe we'll learn from our lesson, we should just have Julie here watching the temperatures. Okay, ladies and gentlemen, we thought it was quite cute. We've done our best to excite Julie Huber about this, but she says nope, 35 degrees isn't doing it for her. So what we're going to do is we switch to our other camera. If we pan up, we can see directly ahead of us, and just about a meter or two shallower, there is a large cluster of shrimp. Earlier in this cruise, we were so convinced that shrimp only live around 10 or 20 degrees that if we saw lots of shrimp, we weren't interested in the temperature of the fluid flow that was going to result. And then yesterday we saw a little patch in the shrimp. We measured temperature around 60 degrees. We set up to do the sampling and we ended up with 140 degree fluids. So we're not going to let the cluster of shrimp put us off and think that we're actually on the pursuit of interesting fluids today. We're actually going to go measure their temperatures and require the fluid to prove to us we're not interested in them once we've measured their temperatures. So we're just setting up again now. This is a larger cluster of shrimp. I think this was the cluster we first saw as we were approaching the site. As we're learning that a lot of these sites are associated with large overhanging boulders. Casey's just blowing at the screen at the moment, trying to see if he can blow the dust out of his site. And unfortunately, Nereus is joining in, blowing as hard as it can as well, and that's probably not helping matters at the moment. And so we have all our thrusters turned off, but it's very still waters that run this deep. So we're just going to take the time to let things clear away. I begin to understand now. I knew when we came here before with Jason, we saw 3 sites of diffuse fluid flow. We stopped at the shallowest one of the 3, and I wasn't sure whether that was just because we were travelling too fast, but I think it may also have been that of the 3 that the shallowest site was the easiest one to sample. And that's somewhat borne out by the fact that the UK team that came back went to that same location for their work. But it's always good to have challenges in life. And trying to work out what the temperature is with this fluid flow site is before we move on. The nature of how Nereus works is, although we knew we wanted to go to the hot chimlet site early in this dive, if we bypassed these sites and left our thin little optical tether behind us, it would be a tricky place to come back to. So if we want to make sure we're interested or not, now's the time to do it before we go on up to the hot chimlet site, and then we'll continue on further forward uphill to the south as the dive progresses. Okay, once more the dust is cleared. We're now set up in front of this second cluster of shrimp in an area of fluid flow, and once again we're preparing to measure the temperatures to see what kind of conditions they're living in, and from that we can learn whether anybody's going to be interested in these fluids. So Julie and Cindy have been actively discussing the cabin inspections and their preparations today. Friday is cabin inspection day, it's a hot topic. Chris Taylor had a different alternative strategy. He was piloting the vehicle down to the seafloor since 6 this morning. Cabin inspection isn't till 11, so he got off duty just in time to put a post-it note on his door and send himself to bed saying, "I'm busy sleeping." So again, as with the other cluster, we're getting temperatures in the 15 degree, maybe up, not even quite as warm as 20. Is that about our limit?
17.
Yep. I think that's about as warm as it's going to get, Casey, so let's stow the temperature sensor. It's kind of confirming what we knew before from shrimp, that they like those temperatures in the 10 to 20 Celsius range. So we've now eliminated those two as sites we want to study. We know there's lots more diffuse flow sites further up the hill, and so we won't worry about these ones today for our sampling sites. So that's the two other sites that we knew were here of diffuse flow. We've confirmed that they're much lower temperature than the hot chimneyts itself, so they will probably now forever remain nameless. And what we're going to do is move across to the hot chimney site itself. What we're passing by before we get there is the marker, the circular plastic marker was put out by our colleagues from the Natural History Museum. It's a whalebone experiment. Basically, we know that some of the animals that colonize vents and seeps overlap with some of the species that are found on large whale falls. When a whale drops to the seafloor and dies, it takes so much organic matter with it in a single visit, or basically at the time that it dies, that can actually sustain chemosynthetic ecosystems from all the rotting of the flesh, can release the same kind of chemicals in the comparable abundances to what vents and seeps release. And so the argument has been that perhaps whale carcasses can represent additional stepping stones for colonization from one vent site to another, or from one seep site to another. Yeah.
That's the one right in front of me.
That's right. So here we are just setting up at this site. Again, we have a one of our little private dust storms carrying with us. So we're just going to let that settle out, and you can just see the yellow nylon cord rising up to the left of the screen. At the top of that is our marker that we left here when we were visiting in 2012, so it would make it easy to relocate the same site when we come back. So just to explain to you what we're doing as our next operation, we have about a year ago when Cindy and I were running a program for the Noah's Ocean Exploration Program, we were actually running a telepresence cruise where on that case we weren't on the ship and using the internet to send information ashore. We were actually on shore running a cruise with an AUV, pretty much the same kind of science we were doing on leg one of this cruise with Nereus, and we were using that in that case to actually explore for and study cold seep sites on the Atlantic margin. And while we were there, we got visited by an artist, Josh Simpson, who runs a program called the Infinity Project. And what they do is, or what he does is he generates little ceramic or silica glass spheres, or planets as he refers to them. Each one is handmade, slightly different, and if you come up with an interesting place of where you would actually like to put one of those, he will happily show it to you. So Casey, could we hold the sphere up to the camera? What you can see here in Casey's manipulator arm now is the silver T-handle, and attached to the end of it with a rubber band is a little blue marble. We put one of these out at the Von Dam site earlier in the week, and this is the one we've chosen for the Piccard site to put out as a memorable little icon of the world's deepest hydrothermal field. And what we're now going to endeavour to do is to put this down right by the vent site here and get some nice imagery of that before we move on for the rest of the program. Hey Jill, could you log for me? Oh, are you back? Oh. Okay. So we're just switching camera views here so we get nicely zoomed in, and then Casey can see what he's doing. So yeah, so that's the project. If you actually wanted to use a search engine to look this up, if you just searched on the Infinity Project, you'll quickly come to Josh Simpson's website. You can find out how to go about this if you wanted to join in the same program. Basically, you write a nice letter, you explain somewhere interesting you might want to go to, and basically if Josh's interest is piqued, then you might, like us, be the lucky recipient of one or more of these. He actually sent us 2 sites, one to put out at the Von Dam site in memory of Karen Von Dam, that's who the event site is named after, and the other for this site at the Piccard hydrothermal field, which is currently the world's deepest known hydrothermal site. And the way this is working is basically the only way that this sphere is attached at the moment is through elastic bands holding it to the metal attachment. So that's what Casey's doing, is just rubbing it against the seafloor to break the rubber band, and then the planet should be released. There it goes. Nearly. There's one little strand of the rubber band still intact. Apparently the whole project was inspired when the artist moved into a house and found some children's marbles left behind in the backyard, and he wondered what the backstory might be of how they got there. So he thought it would be interesting to go to interesting places in the world and leave versions of those marbles or representations of those, each one unique, handmade, so that if they ever get found again, it will just give people a chance to stop and ponder. I'm not sure what the shrimp will think about it. But there it is, beautifully positioned right in the center of the photograph. Good job, Casey. So I think what we'll do now is let that dust settle. We'll let the shrimp get in close to it. And then we'll play with our lighting to make sure we get a really nice image of this before we move on. This is very much the way I'd envisioned it. We actually chose this dark blue marble and we chose this site because we knew about the red-orange coloration of the seafloor here. We figured it should make for something particularly picturesque. And you can see there on that scale, if you think that shrimp is probably at most 5 centimeters, then we can say that this is probably 5 or 6 shrimp lengths. It's probably about a foot away from the hot chimleys vent site. That's a beautiful picture like that. Can we pan a little left? That's great. And if we can maybe sharpen the focus on the marble. Yeah. Use that as a... We've noticed in the past, whenever Julie puts her microsampler on the... Yeah, they got a new neighbour.
I wonder if the heat's different, that's why they were attracted.
Well, it may have retained some warmth, do you think, from when it came down from the descent? Or they're just nosy shoppers and they haven't seen blue before, probably. Well, you know, when was the last time we had an art installation on the seafloor of the Cayman Rise, apart from Jeff's titanium sampler that we put in for 4 years? It's the latest new gallery, there's something new to look at. With the new cameras we have on Nereus, we have two that are different kind of fixed zoom. We have the zoomed in view we're using right now, and then we also have a wide angle view, so we'll take some time to look at this in the zoomed view. So Casey's just started finishing centering up things the way he wants them, and then we're going to start adjusting the lighting. We have multiple different lights on the front of Nereus. We have control of the iris on the camera as well. So there's multiple things we can do to adjust this. And it's kind of nice that Cliff just walked into the room now. Cliff was the guy that helped dial in the cameras yesterday and get them doing nice things for us. So having got the cameras dialed in yesterday, it was perfect timing that we should actually do this art installation project today. So with a very tight focus on where the focus of the flow is, we're doing one zoom shot like this for 60 seconds. What we're going to then do is pan left so you don't quite see the fluid flow so well in the picture, but we'll have the marble right in the center. And we'll do that a second, and then we'll switch to our zoomed out view, and we'll see where the marble is compared to the larger hot chimneys vent site. Okay, so thank you, Casey. That's the end of that sample. Leighton will now pan and we'll set up again, and I'll say when for the start of the next sample will be when we have the lighting and everything just so, with the marble at the center of the picture, complete with shrimp. And you can just see a little bit of the shimmering flow in the very right hand edge of the image there to show how close it really is. Casey Machado, Expedition Leader and Controller of Planets. Okay, so at 1433, let's start that. We'll just hold everything steady with this lighting. This will be Nereus sample 005. Thank you, Casey. Leighton. Yeah, worth persevering. So we've got some very nice imagery there of where the marble sits compared to the vent site. We're now going to switch to wider angle field of view, so we can see the marble. So we're just going to frame that one up and we'll be back online very shortly. So Casey's just going to scooch back about a meter or less. But what you can see here is in the center of the picture pretty much is the blue marble we've deployed. It's exactly halfway between the hot chimneys vent on the right and our marker on the left, with another diffuse flow site, the hot chimneys number 2 site, beyond it. Guess if we go back too far, Casey, the danger is it's going to disappear into shadow.
Oh, we have lights for that.
No, there's some sponges and things around here, other things like that. No, sadly there are no tube worms at this site. The tube worms are all at the Von Dam site. Yeah. So we just got a nice little frame there. We worked out there wasn't a way with a wide angle frame to get everything centered up and not have the yellow of the hull in the view on the right hand side. But then we realized it matched nicely to the yellow of the nylon cord from the marker on the other side. And as any fan of Thunderbird 4 will tell you, you can't have too much bright shiny yellow. Though, yeah, let's drop our Doppler target here for hot chimneys just to make sure we know exactly where we are. And at the risk of being very boring, Casey, I am going to ask if you moved a little bit there, is there a chance that the zoom-in view would have changed at all? I don't want to do it just yet.
I mean, I have edge back, so.
Yeah, so it might be just, that's another 2 seconds. Oh, sorry. Well, we were within 2 seconds of having a minute, so let's call that done. End of that sample, thanks Leighton.
I put the basket in the last second for scale.
Right.
Yeah.
Yeah, it's a good experiment. So, yeah, if we could we, if you don't mind, if we could just switch back to the zoom-in focus. Sorry, zoom-in camera one more time. I think the lighting might be slightly different orientation. I think the marble's straight down from there. There it is. Oh, that looks gorgeous, doesn't it? Now, can we pan a little to the right, please? And then can we bring it up so the marble's at the bottom of the view? Because I think we might just get that upper diffuse flow site in the same frame. Up a little further. It's not quite going to go... Oh, it's almost there. Can you see? Just at the very top of the screen, you can see the lip of that diffuse flow site. And the subtlety of control of the pan and tilt is not going to quite let us have that, is it?
Well, we'll get there.
It's one of the interesting things about the RV Falcor. It's the first research ship I've ever been on that actually has original or has artwork throughout the ship in our cabins, in the corridors. It's quite a nice touch. I know that when I first moved on board, I get the luxury of being chief scientist. I get to stay in the cabin called the President's Suite. The Schmidt Ocean Institute, other sponsors of the Falcor, and Wendy Schmidt herself made a personal intervention that she thought there should be more bright colours around on the seafloor. Or sorry, not on the seafloor, on the research ship. But our contribution is to mimic that and take it one step further. And so that's a very beautifully framed shot. We have our marble, we have the yellows and ochres of the extinct sulfide deposits. On the left-hand side, the shrimp in their fine white. And then the even brighter white of the fluid flow at the very top of the screen there. You can see the hot shimmering water from Hot Chimblets number 2 coming out, as well as the blue from Josh Simpson's marble. So we will start that sample now. And we're going to sit here, I think, for about 90 seconds and collect some of this. Time for Casey to do his yoga stretches. We'll just handcuff him, put him in his special t-shirt. And because every good pilot hates to be in park. They like to be doing stuff and going places and finding new things. And that's what we'll be doing very shortly. And in fact, what I am going to ask Casey is if you pan down in 5 seconds, so you can start getting your hands ready, is whether we can just then bring the marble into the center of field of view and get it the last shot where it's focused up as sharply as possible. So just pan down. There we go. That's all part of the same sample lake, and we won't bother to rename that. Hmm? That's about as pristine as we can get, I think.
You want me to try turning the contrast or the iris down a little bit on this?
Let's leave
Get a little bit more.
this, yeah. We'll leave it another 45 seconds.
So it's kind of washed out. Still have enough for
Okay, and we're just going to adjust the iris a little. Or a lot. No, it's all part of the same sequence. Yeah, well, you know what? Let's catch both, and then we can find out whatever gets recorded. We can then work out in replay. It's like the HDR approach to photography, right? You set it in multiple apertures, and then we'll let somebody go to work with it later on. So, 60 more seconds like this, and I'll be a very happy camper.
See this being one of those motivational posters that says like perspective. Your world isn't as big as it should be. It's as big as you think it is.
Or you're not as big as you think you are.
Yeah, something like that.
We should have had a photo with you holding it before we deployed it. So you'd have Casey Machado with the whole world in his hands. And the shrimp still like it. They haven't gone off. They're still trying to work out how to play volleyball with it, though. It's a bit heavy for them, I think. We'll have to take a little oil-filled one next time, take some carnation oil down and put that in the ball. Okay, that's another 60 seconds, so I'm very happy with what we've just achieved. Thank you very much, Casey and Mario, for making that happen. Leighton for the logging, and Jill for artistic direction from the back of the room. Our aesthetic guru. So, art is what Jill probably does best, but the other thing she does best is sampling fluids, so let's go do that as well. What we're going to do now is we're going to go head for where there are some new fluid flow sites. I would actually like us to try and collect this as fly-through video as well, what's coming up. So we'll... Not just yet, but it's going to be. Yep. So let's come out to the wide field view, please, on the camera. So the plan is going to be that basically this whole hillside from here on upward to the top of the mound is alive with diffuse flow of different temperatures and shapes and sizes. So if we pan up with our camera, I can describe to everybody what's about to happen. And you can see the first of those patches, there's basically a cliff face directly ahead of us, covered in shrimp. To either side of it is more cliff face. You might even need some more light on to be able to see it. But basically it's more of these sulfide overhangs, but if we head off in the direction from here to that set of white shrimp, if we head up and over there, we'll basically find more and more patches of flow. I think probably a little bit over to the right, so we'll basically aim, when we set off Casey, we'll want to have that column of shrimp right in front of our headlights. We'll go up and over there. We're not going to sample that one. It's kind of in a horrible overhang, but we want to have that. We'll film it as we fly over it, and then we should come to a whole world of attractive flow. We don't need to go too quickly because we want Jill to have the opportunity to see different sites. What we're going to be looking for are cues, like if you're looking at these sites now, you can see at the very bottom of the picture in the center, that bright white color doesn't come from biology so much as from mineralogy. It's areas where the minerals are freshly precipitated at these 150 to 200 degree range temperatures, we think. So that's where the hottest flow is. That's what Jill's most interested in. And in and around those areas are places that Julie's also interested in for microbial flow. And they come thick and fast once we get up over that lump. So we'll get our cameras and lighting going nicely, and when we move off Leighton, we will call this a new station for the video. It's one of the most scenic journeys I've ever made in real life or virtual reality on the planet. So it's kind of interesting, I was talking about the scenery of this area. It was just before I came and dived with an ROV on the Cayman Rise the first time. One of my birthday presents my wife bought me that summer was the 50 greatest journeys on the planet you can make. And I actually did 2 of them on that cruise because we actually sailed from Panama. So I actually got to sail through the Panama Canal, which is something that my grandpa used to tell me about back in the day when he was a seafarer before me. And then at the end of the cruise we ended up in Key West and drove the length of the Florida Keys, which is also a pretty cool journey. But what you guys get to do now, I'm afraid I'm not going to pay for you to come on any of those other journeys with me in the future, but this one's pretty cool, intermediate. Halfway between those 2 journeys is a new one which you won't find in the magazines, or maybe you will in the future. Certainly you'll be able to find it online on YouTube forever because we're about to go record it and save it for posterity. But we're basically going to fly forward over these sites, past the shrimp, and work our way up through some pretty cool and spectacular scenery. They're not the hottest vents in the world, but because they sustain a whole amount of biology, they really are quite gorgeous. So if you're a mining company out there, please try and ignore all the mineral deposits that are in the field of view, and enjoy the spectacular life forms that are inhabiting this part of the seafloor. Okay, everybody, Casey is he's about to drop the clutch. Wheels are spinning, we're burning rubber, and we're about to move off. And let's call this sample 009, I believe, Leighton. Oh, you are right. Keep him on his toes. And that's a good sign, so even Sean will enjoy this in the aesthetics, because number 8 is his favourite bottle. Do we have number 8 on the basket today? I do think so. I think we have 7 and 8, so an auspicious day. Lucky number 8. And then I'll throw down the real gauntlet to Casey that Jim Varnum could never fly this route without bumping. Now Casey's really motivated. He's got a completely different one, but if those of you who've been reading our blog, you'll know that that Nereus' stage name is actually Bumblebee from the Transformers. It's specially designed to go into dangerous places where other robots wouldn't go. It's always trying to impress the other robots, and it's specially suited for underwater work. Quite so. Flying right over hot water isn't the smartest thing to do with a delicate little robot. You can see now why it was we didn't try and go and sample around the base of that shrimp cliff, because it would just be a huge overhang. It would give both Nereus and Nereus' pilot huge headaches. Just keep... Yep, and I would, if we can just hang out there for a moment. Let Jill have a look around, see if there's anything she sees that she's interested in. And in fact, if you can steady up there, I might also go and find Julie, or perhaps Jill, you could just fetch Jill in. Jill, if you could fetch Julie from the library. Some of these other sites with the shrimp did get sampled previously by Julie, but the microbial flow, the microbial activity, of course, is restricted, as far as we know, to temperatures of 121 degrees and lower, whereas the fluids here can go up to 150 or 200 degrees. We can see where we were just now. We were in an area with a lot of orange-red metalliferous deposits, a few shrimp around and some active flow. Here what we see are much higher concentrations of shrimp, which presumably are not linked to less vigorous fluid flow. But we have Julie and Jeff as well as Jill now back in the control room, because we're now entering the area where they want to sample today. We probably might end up being here for some time. So as the light becomes clearer, what you can see in the lower right of the picture is a large white area teeming with shrimp. Now we know earlier today when we tested fluid temperatures around where the shrimp were happiest, the fluid flow was only around 20 or 30 degrees Celsius. But what we can see right at the bottom of our picture is a little dark area in amongst where the shrimp are. I suppose it could be. So the danger is it's just a is that just a shadow being cast by the ridge from above, or is it? If it's actually a little crevasse, then that could actually be quite hot flow in there. Is it worth switching to our zoomed camera rather than before we? Okay, I was just thinking rather than try and get in there, because if that is an overhang and it's just casting a shadow, then we're going to cause ourselves lots of struggle for no reason. Yeah. Then if we... Yeah, I think that's just, it's casting the shadow, isn't it?
Yeah.
So that's actually, yeah, it's not a black crevasse after all. So then I can definitely see active flow at the base of that pile. So we could just nudge forward into, can you see there's like Schlieren effect between us and the shrimp? I think that's really, I think that's just dust, is it? Yeah, because if there is an active flow in this foreground part, then that's not going to be the kind of temperature range that's going to be of interest. I can see some bright white, that little patch, but that's going to be a stupid place to try and get to.
I agree.
But that's the kind of colour we'd be looking for in that little one, if you see those bright white spots.
Bump up the iris on fire there.
I think I can see something just there. See that?
Yeah, yeah,
That's like a grey discoloration. Conceivably. Yes, that'd be a great idea, thanks. And Julie, since you've got more samples or more sampling capability than Jeff and Jill, should we start with your temperature probe for this site and see how it looks for you? Because they can only sample one subset or one or two subsets of the places you're interested in. So if we measure it and the temperature's good and you're happy with it, we'll take your sample first, and then if temperature... A sample.
No, just ping it here. I've already called this one.
We can call it the last CTD. And then we'll make Max honor that commitment. So we're just settling up now, and we're going to get ready to sample temperatures here. So, we are about to have a great time here. We are in the joys of telepresence. We actually have a so you can hear lots of grown-ups being very bullying in the back of the room here at the moment. A lot of hilarity. But one of the programs I've been working on at home is developing the use of telepresence and engaging early career scientists in that. So that's who's going to take over the microphone for the next while. And we're going to start with Julie Revaillot from MBL, who's going to come and discuss what's happening as we take these temperature measurements and then potentially migrate into sampling for the work that she's been doing with Julie Huber, otherwise known as Jules Deep, or with her new branding for the University of Chicago, allegedly going to be changing her Twitter to Jules Deep Dish. And so with that, I really am going to have to give up the microphone and be taken off kicking and screaming to my cell. And the next voice you will hear from will be Julie Revaillot.
Good morning. So my name is Julie Revaillot, Julie 2, as I'm called here on this boat. I work at the MBL in the laboratory of Julie Huber. We are both doing some microbiology of deep sea vent fluids, and on the simians of the tube worms you have been seen in the Van Damme field that we were studying yesterday. So we are now at Piccard field, trying to look for the right sampling site with the right temperature, trying to get as close as possible as hand members temperature fluids.
What's the temperature? 5. We're nowhere near it yet.
So temperature is 5, I heard, and we are getting closer to a diffuse flow sampling site. So for my French colleague and for people speaking in French around the world listening to us, I can try to speak a little bit in French. So bonjour à tous, on est sur le site de Piccard, sur le Mid-Cayman Rise. au sud de la Jamaïque et de Cuba. Et on est en train d'échantillonner les fluides, donc les sources hydrothermales les plus profondes du monde. On est avec le HROV Nereus de l'Institut de Wuyi. Donc on est en train de faire des mesures de température. La température actuelle du site que vous voyez sur l'écran est de 30 degrés Celsius. Et on sait que le N-member va jusqu'à 396 degrés. C'est redescendu à 10 degrés. Donc autour de nous, on a les crevettes de l'espèce Remicaris hibisae. « 8 degrés. Donc on est à bord du navire océanographique de Falkor de la Fondation Google, avec 9 scientifiques à bord de l'Institut de Wuyi et du MBL. Toute l'équipe du Nereus. Chris German est le chef scientifique. Donc la température est toujours basse, à 7 degrés. La profondeur actuelle est 4982 mètres. Donc Cindy Vandover, qui est à bord aussi, a commencé à disséquer des crevettes qu'on a pu échantillonner dans les plongées précédentes. Et on a trouvé hier que des snails, donc des escargots, et dans l'estomac des crevettes. Donc c'est des crevettes qui vivent en symbiose avec des bactéries chémosynthétiques, mais on vient de découvrir que c'est aussi des crevettes qui se nourrissent d'autres organismes. La température est 18 degrés. 21. Oui. Yes. Donc Chris German a repris le siège des commandes. Et à ma droite, il y a Leighton. Hello. Qui est en train de rentrer toutes les données sur la plongée pour avoir accès plus tard au log. Donc pour mes collègues américains, je vais peut-être repasser en anglais. What? So we are 21 degrees now. And there are still thousands of shrimps surrounding the diffuse flow. And we are trying to get the more stable temperature of this site.
Well, I went all around it and I didn't get any temperatures that were not all the way back.
So we are now at 26. I'm trying to stay there. 41. So people are getting excited. It's getting high.
Did you say 41? Yeah. I did. Now I'm saying 49.
49.
Nope.
I did bump it to the side there.
I think maybe bumping the side is good.
Bumping the side.
Well,
is good.
what is it right now?
30.
Still
Okay,
unstable, but
Bring on that left side. How about
36,
Okay, chip.
42, 43.
43 degrees. So as soon as we get a stable temperature, I think we're going to sample some water sample for some cell counts and fish experiments of microbes, which is mainly fixing the microbes into a stage that we can later on count them with a microscope and use some probing of different microbial taxa to see how they are located and in which quantity. And then we're going to use some Sterivex filters through a pump to get as many as DNA as possible and extract this DNA and amplify it later on and try to get an idea of the diversity of the microbes that are thriving in these deep sea vents ecosystems.
That looks pretty good. Sean's making them up. Great. This way.
Is that 56?
Yes.
All right, can you lock it there, Casey? Is that
So we are looking now.
thanks?
We are going for sampling
Grab and chip.
through a sampling device called the Super Sampler that Chip at HROV designed and is managing here on board.
Drill, baby, drill. It was a very silly English comedy show.
So the hand member is about 112 degrees, so that's why we are interested in to this 56 degrees temperature fluid, just to have an idea of a temperature in between. So the arm is now fixed on the seafloor, and we are initiating the software to start the sampling. So meanwhile, people are laughing behind. They are having a good time, lacking of sleep, I guess. Today it's the cabin inspection, so everyone has to clean his own cabin, and the crew is coming and giving us some smiley face if we did a great job in cleaning our cabin, or a sad face, or a note saying like try better next time. So then we all compare our stickers and we can make some exchange. Like, so this will be a surprise. Like in half an hour, do we have a great face, happy face, or not? This is the deal of today.
We're starting to purge.
So we're now starting to purge. I heard at a 56-degree fluid.
And then Jill, you'll take over? Or are you going to take sample here? Okay.
You're gonna what with the who?
Don't ask. It's a little bit dirty sounding.
Maybe more cylinders.
I'm happy with this.
Yep.
What's the sample number again? 009.
This is 9, it's a continuation of 9.
So in the control room there is Chip starting the software to pump. There is Jeff measuring the temperature. Mario and Casey are driving the ROV. Julie Huber is next to me on my left, trying to get to know about the best accurate measurement of the temperature. And Leighton next to me is taking notes of what is going on and filling the logbook. There is Jill also, and Sean, sorry. No, not Sean. Who are measuring all the chemistry of these vent fluids, the magnesium that remain, and the pH, the hydrogen sulfide concentration, the methane concentration, CO2 concentration. And so there is a very nice collaboration between the microbiologist and the chemist here on board.
Whole water sample.
So we are sampling the water from this vent fluid at port 1.
What's the temperature?
And the temperature is now 23.
What are you pumping at, Chip? Okay.
It was 73 when we started.
So we are pumping slowly in order not to get as much as seawater as possible. And this can last 5 minutes, I guess, for a water bottle of how many liters is it?
It's a liter bottle.
A liter
Trying to
bottle.
Flush it 7 times.
So it's a 1 liter bottle, and we try to flush it a few times.
It's sort of a weird thing in science.
I've heard that if you triple shuffle a deck of cards more than 7 times, you're no longer randomizing the cards.
You're getting back where you started.
Or you start to introduce a pattern into it. I don't know.
So the shrimps seem quite happy here. The temperature is not too high, apparently. They're all happy.
Can you tell me what the temperature is?
They are really dense. There are millions of them. Maybe not here, but from what we have seen so far, 1,500 per square meter. This is an exact data, apparently.
Very stable. Stable at what? 15.
So the temperature is stable at 15 degrees. So there are a lot of microbial mats at Piccard, where they are not at Van Damme, which shows that the chemical and physiological conditions, environmental settings at both sites are different in many ways. So for a description of the Falcor we are on now, it's a very nice boat. We have access to the log, like from each room, from the kitchen. There are screens everywhere where we can follow where is ROV, what is going on the seafloor, what is going on the deck. Food is amazing. There is a sauna, which I didn't use yet, but I guess some people did. There is a gym room.
Sauna?
Yeah, I said there is a sauna. Sometimes there is a yoga training session on the top deck with a view on the sea. There is a nice lounge for scientists as well as a nice lounge for the crew. We have the luxury to have someone doing our laundry, which is like really appreciable. And well, it's all well equipped and very comfortable. So we are sampling at 4,993 meters at Piccard. The site calls... How is the site called again?
Where are we? Where are we?
So we are not far from Hot Chimney, but the site don't seem to have a name already. So the
Near a
Japanese...
place called Hot Chimney.
Yeah, we are at a place near Hot Chimney. So our Japanese colleagues are also around, diving with their submarine Shinkai. So this is a lot of data collected during this month, and I guess it will be
Stopping that sample at 1528.
doing some great science. So we are stopping our sample after 5 minutes of pumping. Temperature has increased slightly
Let me know when you're starting number 2.
to 64 degrees.
Here is 10 bottle port number 2. Yep, starting to
Starting 2 at 1528.
just stay way back.
Starbucks.
So now we are starting another sample, which is a Sterivex sample.
Now you're stable.
So we are pumping through this filter to get the DNA and the RNA that we can extract and amplify later on. And as I said, like to get the diversity and the functional genes of these microorganisms. So for this particular sample, we take a longer time, like we sample for about half an hour,
We'll probably do closer to 20 minutes. Just
or maybe 20 minutes only, because the time is limited here. So we are at Piccard, which is really deep, which with not so much time on the seafloor that we have to share between all scientists, and then come back to the surface. So we are trying to organize the dive as much as possible and share this precious time so that everyone gets some good samples and good data to analyze back in the lab.
we'll go on vacation when we get home, Casey.
People are now speaking about vacation time. People are dreaming about a nice, good night sleep of 8 hours in a row, not
Yeah.
4 hours and then 2 hours and then 4 hours.
It's not really pumping through it.
But an amazing fact is that people, even with lack of sleep, remain really nice and smiley. Until when, I don't know, but so far it's going really well.
How can you tell?
Well, I'm looking at the photo.
Oh, okay.
So the temperature is 60 degrees, and we are getting through our Sterivex sample. So when the ROV will be on deck, we'll get this sample, put it back in a bigger tube, remove the air and the liquid that is there, push it, and then fix it with some salty solution called RNA later. Put it in the freezer at -80 until we can extract the DNA. That's the way we do it.
Clayton, do you like our USB L-Fix scatter plot?
Well, this is me. Oh, your one. Oh, yeah. Actually, when you think it's within 20 meters, and we've got a cluster here, so what I'm doing is later on, if the boat
Drop
is
the CTE on the
is
cluster.
drop it in the middle of there. They all point into generally this area here, I'd say.
So a lot of CTD samples have been done during leg 1 and some during leg 2
We get good results with this method. That's a real good spike.
to get to know the chemistry of the plume water samples and of the background seawater. Max on board is studying the lipids that he found in these seawater samples.
That would be so
A colleague was trying to get the virus on these samples. Cody. We believe there is a lot of interaction between this virus and the microbes, and that they play a huge role in the evolution
pretty soon you'll be able to like
of microorganisms.
watch them on TV. No one will ever have to go to sea again. I can't wait until I can like show up at work at like 8 a.m., flip on my computer, pull out my little joy box.
So we are sampling and sampling, and
pilot, have a little
can
headset,
take some time
Yeah, no, I think that's
for these Sterivex samples, and so
that would save, and then you just have like 3 people or 4 people on
people are having some nice discussion in the control room.
Have a drone which can even fly the gear out there.
voices.
So Leighton, to my right, will now take over and tell you more about what we are doing and what we are seeing here at Piccard.
Hello, the reason I've been put on is because by all accounts a British voice on the telepresence sounds good. Unfortunately I'm Welsh, so my accent's a little bit off, so Borida to all of those in Wales. No, it is, I'm on the other microphone. So at the moment we're just sampling for Julie. We've been sampling, is it for about 10 minutes already?
Yep.
10 minutes. We're just drawing some of the fluid you see from the vent site in front of us into a filter, which then I think Chip and Julie will process when it comes back on board. At the moment, everyone's just sat in the control room. Casey's with the manips. Julie's updating Twitter. Jill, whose family are probably watching as they're avid followers, is sat on the other side of the room making notes. Chip's staring at his temperature probe. And apart from that, there's been a rather reduction in people in here in the last 5 minutes, namely because it's lunchtime and our chefs, as usual, are making some great food. It's like a tide during meal times in here. Everyone just moves out and leaves the important people in here.
Yeah, that's it.
For friends in the UK at the National Oceanography Centre, I notice now it's getting close to 3:30 in the afternoon in the UK. I can imagine we're going to be losing a lot of you soon, as it's Friday and the platform tavern's nearby, so thank you for tuning in. And if any of you are looking for something to do tomorrow morning, tune in and watch us on is it last dive tomorrow or penultimate?
Not sure.
Okay, there may be more. So I'll give you an update on the rest of the activities. Once the Nereus Dive is finished, we expect that to be around 6 o'clock this evening. We're going to switch back over and do some CDDs for Max. I'm not sure on the site at the moment, but it may be somewhere in the vicinity of the area we're sampling at the moment. One of the good things about sitting here doing this sampling for a long period of time is we can actually fix the location of the vents using our ultra short baseline system really accurate. Later today, if we do CTD here, we can now get the position of this vent to within a meter and a half, and hopefully get some really accurate samples with our CTD. We've used this method the last few sites and got some pretty spectacular results. while we've been sat here, we've actually noticed on one of our other cameras that in front of us is quite a lot of smoke. I'm actually just going to turn over our cameras.
Here, see, smoke.
You can see in the background there's some smoke. Once we finished here, we might go over and take a look and see what the source of that is, whether or not it's another smoker or some campers. Currently there's an ongoing debate whether or not this is actually smoke or just sediment which has been stirred up.
All the way down to the firebox, right?
Casey believes it may be smoke coming from,
It's not smoke, it's crack.
sorry, dust coming from the crack in front of us.
Yeah, it's just
Particles.
particulate
Particles.
in the water and the crack and really bad video tonight for cameras. All part of the illusion we have to create the safe the seafloor here. Used originally.
Okay, ladies and gentlemen, we have approximately 8 minutes left on this sample, and then we'll be doing some more sampling. So during this period of sampling,
Pardon?
oh, sorry, no, I said during this period of sampling, Casey is going to give us some nice images from around the vehicle. As you can see at the moment, he's zoomed in on a nice cluster of shrimps. You can see the shimmering water in which these shrimps are living. And that coming into view here is the vehicle's manipulator. Moving over to the right, we have more shrimp clustered around some more diffuse flow. Seems to be a reoccurring theme here with shrimp. There we go. Earlier I was speaking to Cindy and she actually did an algorithm and lots of counting where she said that there are roughly, in some of the other sites, 1,500 shrimp per square meter. As well as supporting shrimp, you can see the fish we often see. I think these are zooacids, not at this site but at the other ones. And the occasional crab or swat lobster, galatheads. At the moment, Jill's just coming in because Julie's off for lunch, so I might actually hand over to her in a minute because I know her family are avid listeners. I had an email last night from one of my friends asking how Nereus's manipulators survive the heat coming from the hydrothermal vents. Well, that's due to the 6 figures that it costs. But they're really reliable, rugged, and expensive with it. But they can easily survive the temperatures that we're encountering here. Apart from as well as designing arms for subsea vehicles such as Nereus, the Jason ROV, ISIS ROV. I believe Craft also developed manipulators for use in nuclear power plants and also on bomb disposal vehicles. Subsea manipulators is just one small part of Craft's manipulator market. Another big shout out to Lee Marsh. I believe she's watching, but I hope you're enjoying the footage. Okay, I'm now going to hand you over to Jill because it's lunchtime for me, and Jill's going to be entertaining our 30 plus viewers now, plus her family.
Good morning, everyone. This is Jill McDermott reporting live from the Mid-Cayman Rise. We're sampling at the Piccard vent field this morning. Nereus is our ROV, currently at a depth of 4,992 meters. So this is the deepest vent field on Earth currently known. And what we're doing right now is collecting a microbial sample for Julie Huber of the Marine Biological Laboratory in Woods Hole, Massachusetts. We are pumping right now for about 7 more minutes to collect a whole water sample, so about 1 liter of water of this hot fluid which is leaking from the seafloor. And I am actually also based in Woods Hole. I'm a doctoral student in the MIT Woods Hole Oceanographic Joint Program in chemistry. And I'm working with Jeffrey Seawald and Christopher German, who you've been hearing a lot from. And this site right here is particularly interesting to me because this is one of my thesis sites of interest. And I'm a vent fluid chemist. So we're at a site here called Hot Chimlet. This is at maybe about 100 meters away from the BB vents, which are the spectacular looking high temperature black smokers. Those have a fluid temperature of 398 degrees Celsius, very hot. And This hot chimlet site is interesting because we have this very broad temperature range that we're able to sample. And in particular, this 100 to 200 degree fluid temperature range has not really been well represented in sampling efforts worldwide. This is a rare type of fluid to see on the seafloor. Usually you get either very hot fluids, something around 350 degrees to 400 degrees, or something a lot cooler and very well mixed. It's more like 20 to 50 degrees. Whereas here, this fluid is, we're measuring maxes around 70 degrees in this spot. And so earlier this week on Tuesday, we were about a couple meters away and we sampled a 130 degree fluid. And what's interesting about that is we're able to cut across the abiotic-biotic temperature boundary. And I say that because the known limit for life presently is set at 122 degrees Celsius. And that was determined by our Japanese colleague, Ken Takai, in his lab in 2008 when they grew a hyperthermophilic methanogen. It's a type of microbe grown under very high pressure and at a temperature of 122. So we know life can survive at 122 degrees. So one of the questions I'm looking at during my doctoral work here focuses on the difference between fluids that are cool enough to host life, so less than 122, and fluids that are just over that temperature, so 130 degrees and hotter. And then comparing those fluids with the very hot black smoker fluids, which are about 100 meters away. And so once we finish this microbial sample, we'll be collecting what's called an isobaric gas-tight fluid sampler, which are the types of samples we collect in my lab. And I'll discuss that a little more when we get there. All right, and we are now stopping this sample. We've just collected a whole water sample for microbial life, about a liter of water. And the temperature in this sample was around 55 degrees during sampling. And we're setting up now to sample with our isobaric gas-tight samplers, which will collect a sample at pressure, which remains at pressure. And so we'll be able to investigate the gas chemistry in these fluids. But what's going on right now is my thesis advisor, Jeff Seawald, is sitting next to me in the control room, and he's trying to convince the pilots to excavate the seafloor a little bit here.
No, it's fine.
With the manipulator arm, just to give us a better conduit for fluid flow, to see if we can tap into the hot fluid that's just below the seafloor here. This is a hot discussion going on right now. whether or not the seafloor is soft enough to actually impact, or whether or not we should just kind of take it as it is right now. I think we are going to try to scrape at the surface a little bit.
So are we done with this?
This is Jill McDermott coming to you live from the RV Falcor, sampling at the Piccard vent field at the Mid-Cayman Rise. And we've just completed some microbial sampling at this site. And what Casey, our ROV pilot, is doing right now is some basket maintenance. He's moving this net around so that he can get a better handle on our next tool
Hey Jill,
that we're going to be using.
your bathroom cleaning earned a picture. It was so nice.
Oh my gosh. I just heard that I've passed my bathroom inspection with flying colors. I earned a picture on my post-it note this morning. So that is really cheered me up. I went on a cleaning frenzy at 2 a.m. this morning, which is not very long ago. Right, and now we're picking up our next tool, which was previously used this morning to deploy some artwork on the seafloor. We put down a planet glass sphere, which was made by Josh Simpson, who's a glass artist working in Massachusetts. And we had this handy little tool for placing the sphere, and we're going to now pick that up and repurpose it in order to scrape at the seafloor here to see if we can make ourselves a really good orifice or a conduit for the fluids to flow through in order to sample the most pure vent fluid we can. At the moment now, we are picking up our next tool. It's just to the right of your view here. We're on our zoomed in camera so that our pilot Casey and Mario can get a good view of the tool. It's got that yellow tape on the end of it for better visibility. Here it comes. It's a pretty delicate maneuver. It's kind of over in that corner and hard to get.
Oh, you do. Into the bio box. Panned over. It's in the bio box. I might not be able to get it. Is it really in the bio box? It's kind of, I don't see it
The
at all.
tool is eluding us.
Go ahead and mark to the side.
That other box just to the right is our bio box. That's our. There we go. Oh, close. The box over to the right is the bio box, which is where we would put our biological samples if we were to collect any shrimp or anemones, or if we happen across any mussels. That would be very exciting. So that's what we're referring to over there. And the tool, this was the art deployment tool.
Okay.
Yeah.
It's just in the way at the moment.
We're trying to pick it up so we can scratch at the seafloor with it
Oh, okay.
and make a better channel for the fluids to flow through. Good afternoon, everyone. This is Jill McDermott. I'm live in the science control room on the RV Falcor, and we are at the deepest vent field on Earth, the Piccard vent site, preparing to scrape around the sediment with this tool that Casey has just grasped with the manipulator arm in order to select a site for sampling hydrothermal vent fluid.
Alright. Let's just bash away with this. So where we were before,
Yeah.
just bigger and better.
All right, we're all set up now to to gently scrape at the seafloor here.
Alright. Zoom in, scraper destructive mode activated.
We're switching cameras to our high zoom camera. This is a high definition camera donated to Woods Hole by James Cameron from his deep diving submarine. And we'll be able to see a nice view here where the fluids are leaking out of the seafloor. This shimmering flow, wherever you see the most shimmer, that's usually where the temperature of the fluid is the hottest. And this indicates that that area is where the source of the fluid is coming from. This is the most pure vent fluid, and that's what we're interested in sampling within the next few minutes here. And Casey is our running the manipulator on Nereus. He's seated 2 chairs over from me in the control room, and he is preparing to use this tool to excavate a little bit where we just took our last sample to see if we can create a little more flow. This is Jill McDermott. We have completed that excavation, and now we're going to pick up the first of our gas-tight fluid samplers. We're going to get the rearmost sampler, which is the one at the bottom of your view right now. That one's bottle number 7. And we'll be taking our first gas-tight fluid sample from this location in just a few minutes. thank you for joining us at the Mid-Cayman Rise. We are just clearing away some of the other tools in order to get a good grip on our gas-tight fluid samplers. Just moving that blue net over. And then we'll be collecting our first fluid sample for gas chemistry. Good afternoon, everyone. We've just picked up IGT number 7. This is a gas-tight hydrothermal vent fluid sampler designed by my advisor, Jeff Seawald of Woods Hole Oceanographic Institution. It's firmly grasped here by Nereus, and we are about to sample a vent fluid at the deepest vent field on Earth. This is the Piccard hydrothermal field, and we're at a vent site called Hot Chimlet. This is a really exciting fluid for me because I'm doing my doctoral thesis here, and we were just, not at this site, but very close to here last Tuesday, and we sampled a fluid there that was 130 degrees, which puts us just over the temperature limit for life, which is currently set at 122 degrees Celsius. And this fluid is on the other side of that temperature limit. We just measured a maximum temperature of 70 degrees during our microbial sampling just about an hour ago. So we're going to try to collect a vent fluid here in order to intercompare our results with those of the microbiologists. And this is a really exciting temperature because we'll be able to determine whether the fluids as they mix in the subsurface are being modified by microbial life. Are the microbes actually consuming or producing chemistry? Or are we simply heating things and pyrolyzing material and altering biomass that's already there, that's being entrained in the hot fluids? And then the third process that could be going on is what we would call an abiotic process, which occurs in the absence of life. It's purely chemical with no life necessary. So disentangling those three processes, microbial consumption or production, thermal alteration of biomass or abiotic reactions is a very interesting question. There's a lot of interest lately in understanding the subsurface biosphere in our oceans, and this is an excellent spot to be studying that due to all this chemical energy that's available for microbial metabolism and production. So what I'll be doing with this sample is analyzing probably 30 different species in it. On board the ship here, we're mostly focused on determining the volatile chemistry, which is what I'll be doing tonight after dinner. When the sampler comes back on deck, we can measure hydrogen, methane, and carbon monoxide here on board. And then I'll be taking the fluid home and looking at other volatiles, including carbon dioxide content and other organic species in the fluid, like hydrocarbons, ethane, propane, very chemically rich, energy-rich chemicals, all of which could be used by microbial life in the subsurface. And right now we're getting live temperature readings from the thermocouple temperature probe, which is attached to the inlet of our fluid sampler, which you can see right in the center of your frame. It's pressed up where the fluid is emanating. Our max temperature so far is 85 degrees. And what we'll do now is sort of maneuver the tip of the snorkel inlet around to find a nice, hot, stable temperature, which is...
You happy with 82, Jill?
Yeah, 82 sounds good. All right, we're holding right here.
2 minutes, right?
We're locking the arm
Stable? I can hold
and
it for 2 minutes.
discussing with our pilot Casey whether or not he's stable. We need 2 minutes, 2 minutes of vehicle stability in order to collect a sample to give the bottle time to fill. Just adjusting it slightly here. And the way we're getting the temperature reading is using something that's basically like a modem on the seafloor. It's in the basket of the submarine. And we're getting that signal traveling up from the seafloor along the fiber optic cable and being transmitted right here to the control room to Jeff's laptop right over in the corner. They're stirring up some biology at the moment. And we'll wait for things to settle down a little bit. We're back in position now, sampling with an isobaric gas-tight sampler at the hot chimlet site on the Mid-Cayman Rise. This is the deepest vent field on Earth, and we are setting up to sample an 85-degree fluid. This is a fluid that's been mixed in the subsurface. We know that what we call the end member, or pure vent fluid at this site, has a temperature of 398 degrees. And this fluid here has mixed with seawater and lowered the temperature down to about 85 degrees. This is a great temperature to find what we call hypothermophilic microbial life, so microbes that love hot temperatures. Our temperature max we just got is 96 degrees, and we're opening the bottle. Starting this sample right now. Stable at 95, 97. That's very good news. And this bottle will take about 2 minutes to fill. We can open it from the control room and then trigger it to close when we're satisfied that it's full. And what you can see attached to that bottle right now are the electronics housing with the cord coming out of it. Just at the top of your frame, where that plate is folded over the end of the bottle, that's the actual area of the bottle that holds the sample. So we take about 150 milliliters of fluid. It's filling there, you can see the end of it now, filling up that chamber with 97-degree vent fluids. And this bottle is constructed of titanium. We choose that because it's chemically an inert metal, doesn't react, so it won't contaminate our sample with any metals. It's also very pressure and temperature resistant. And these samples, when they come back on deck tonight after dinner, will still be at seafloor pressure. So at a depth right now of 4,985 meters, they're going to come back at about 500 bars pressure, which is 500 times pressure that we normally feel, 500 times atmosphere. And what we'll do in the lab is carefully extract the fluids using a high pressure pump to, in a controlled way, get the fluids back out of the bottle. And we'll have both the dissolved gases and the aqueous or liquid fraction, both combined in one sample. And at that point, tonight we'll be measuring the volatile chemistry, the gas chemistry on the samples. We'll measure the hydrogen, the methane, and the hydrogen sulfide abundance, as well as the pH and other unstable chemicals. We like to measure those on board. So that sample's done. We just close the bottle and we'll stow bottle number 7 back in the basket, and we'll be collecting a second sample with another gas tight sampler. And the reason we take 2 samples is it gives us more confidence that we've actually got a good representative sample of the fluid that's coming from the subsurface. So if the 2 agree well, then we know this is really what was exiting the seafloor. So we're just taking a minute here to stow bottle number 7 and prepare to pick up bottle number 8. This is Jill McDermott, and we're on the RV Falcor sampling at the Mid-Cayman Rise vent field. We've just picked up our isobaric gas tight sampler number 8, and we're preparing to take our second vent fluid from this hot chimlet site. We just took a sample at the bottom of your screen that was 95 degrees Celsius. So that fluid we think is about a 1/4 vent fluid and 3/4 seawater, which is a very interesting sample because it's going to give us an opportunity to look at mixing and cooling and how that impacts chemistry of these fluids in the subsurface here. And at 95 degrees Celsius should be a temperature at which microbial life can flourish. And so we'll be able to see whether or not these fluids have been impacted by microbial processes and whether or not that's reflected in the chemistry. And these fluid samples will make up a large component of my doctoral dissertation at the MIT HUI Joint Program in Chemistry. And what we're trying to do at both of these vent fields, the Piccard and the Von Dam vent field, is sample across the temperature range. This is IGT number 8. IGT number 8 is also famous for having sampled the broken wellhead at the Deepwater Horizon Macondo well during the oil spill in the Gulf of Mexico. So these fluid samplers are useful to sample any type of fluid which is under high pressure, whether it's at the seafloor, vent fluid, or petroleum samples. We're now getting temperature readings from our thermocouple, which is attached to the snorkel. On the very tip of the sampler, you can see it amongst the shrimp. We're reading 55 degrees right here, 54. See if we can get that up to 95 like the first sample. .73 in that spot. Looks good. .75 degrees in this spot. As you can see, the temperature is very variable. If you move the snorkel even a centimeter, you can have temperature drop very quickly. So there's a very steep temperature gradient at these hydrothermal vents. The ambient temperature here, the temperature that the vehicle Nereus is sitting in, is around 5 degrees. Reading 62 degrees here. And what we're looking for is a nice, high, stable temperature.
cold to the bottom?
No, it's not as hot there, Jeff.
Not as hot?
No. I don't think so. We're trying a different...
44, 52.
Oh, maybe I was wrong. 60 degrees. Usually, when you look at a video like this, the hotter parts of the image are where there are the fewest shrimp. Because the shrimp can't live in a 95-degree fluid. That's too hot for them. So we're looking for black areas where there are no white shrimp. And hoping that those are the hottest. The area where the source fluid is leaking from the seafloor.
76, 82,
82 degrees,
65,
60 degrees. This is a really small area
69,
where the hottest fluid is coming from. So we're looking for that.
63, one, 41, 50, 70, 54, 20, 31, 46, 49, 51, 65,
We've got
87,
90 degrees. All right, in this spot.
73,
It's our highest reading so far in this bottle. We haven't opened it yet. We're still
82,
trying to position that snorkel right in the hottest fluid.
78, 82. Take it, Jill, or
Yeah. What is it at?
it's fairly stable about 83.
It's fairly stable here at about 83 degrees. It might be about as good as we'll get.
Just drop.
Oh, it just dropped out.
Now we're back at 8.
It's funny. We're not really moving the.
If I give it a twitch. I didn't do it. We haven't moved, so. Yeah.
We are not moving the sampler at all, but the temperature is still a little bit variable. So it looks like we're right on the edge of some flow. So we're seeing if Casey can move the sampler just a little tiny bit. 76 degrees now. Ooh, 90. This is a good one.
I jiggled the vehicle very slightly. Really? Do it again. Okay, hold it right there.
Okay,
Here we go.
what is it, Jeff? 98?
We got to 101.
Max temperature of 101, but now it's back down to 70.
Another, whatever you've been doing. 5 minutes, okay.
So Casey is giving the Nereus a jiggle. It's a technical word for this.
68.
It's now We're probably right on the edge of a jet. So there's some mixing occurring right at the seafloor. So we're interested in getting the least mixed fluid, so that's the hottest temperature. It's pretty amazing that we can do this at all, honestly. Are you opening it? Can we try to get up to 80? All right, we're opening the bottle, sampling. 96 degrees Celsius has been our highest temperature. Now that the bottle is open, we're going to wait 2 minutes for it to fill. And it's been designed in such a way that it fills slowly. And it's under very high pressure, so in order to do that, we've got a very small diameter inlet. Our snorkel is very skinny, like a straw, and that's sipping the fluid from its source into the bottle.
What IGT is this?
IGT number 8. And within the bottle, there are 2 pistons that right at this moment are moving back very slowly. And in between the pistons, there's a tiny orifice called a viscojet. And the viscojet is our flow control. So that allows the fluid to seep into the bottle slowly so we don't slurp it in very fast and entrain a lot of seawater. So we're filling over the course of 2 minutes. It probably takes about a minute to fill. So we need to hold steady for 1 minute here in order to fill up that chamber. We're collecting 150 milliliters of vent fluid inside a titanium bottle. And that's going to come back up to the surface at 500 times atmospheric pressure later today.
What
And
time
that
was the previous sample taken at? It's not noted here.
we've got a rock solid temperature. What is it at, Jeff?
Started at about 100 for the first 25 seconds, and then drifted down to about 80.
Okay. So that sample looked like it started around 100 for the first 20 seconds. The bottle was open. Now it's dropped down to 80. So we might be getting a little bit of a mixture, but I'm happy with 90 degrees. That'll be a really good sample. And the bottle probably fills quickly, so it's probably at least half full by 20 seconds. And we're controlling the electronics of the bottle. You can see them in the top of your screen with that cable going in. That's our cable with which we are transmitting the temperature measurement all the way up to the ship along the fiber optic tether and into the control room where I'm sitting. All right, and that sample is finished. We just closed the valve on the bottle, so it will now hold pressure at 500 bars. We're at a depth of 4,982 meters, sampling at the Piccard vent field, deepest vent field on Earth. And now we are stowing isobaric gas tight bottle number 8. And we've completed our gas tight sampling for the day. Got 2 very nice samples, so I'm very pleased. Sean, do you want to say hello? He's shaking his head. Jeff and Sean's families are not listening, but I know mine are. Hi, Mom. Just a shout out to Maine. My mom, Linda McDermott, has been tuning in. She should have been a hydrothermal chemist. And she's watching live, and so is my boyfriend, Santiago, in Woods Hole, Massachusetts. We've just watched my thesis in action. Santiago? Yeah, he's Colombian. He's from Colombia. He sailed on the Falcor last summer during the shakedown cruise. All right, this is Jill McDermott signing off. I'm going to go eat lunch, and I'm handing the microphone back over to Chris German of Woods Hole, who's our chief scientist on the cruise. Here's Chris.
And Dr. Zoidberg is also leaving the room. So, no, it's not time to go home. It's time to go exploring.
Time to go.
But we're going south.
Going south, and we discovered something.
We already discovered something.
We're always discovering things, right?
Correct. So the time is 12:45 here on the RV Falcor. It is Friday, June the 28th. We are diving on the world's deepest vent sites today, the Piccard hydrothermal field. And we're about to go in exploration mode. We've just been sampling some fluid flow sites just uphill, just south from the hot chimlet site. And we're going to progress smoothly and steadily ahead. I think over to your right a little, in case you're pretty much dead ahead and to the right in the forward-looking cameras. We can see a whole wealth of more
That's actually to the left.
chimneys. Is that to your left?
Well, because the camera's looking from the right side.
Ah, you switched the camera, sorry. This is the one that looks straight ahead. The other left. I didn't get where I am today without knowing where I'm going. Okay, let's hold up slowly there, please, because I want to take a scout around and see where the fluid flow's coming from. Can we pan up a little with your nice camera? Show the nice people at home out of the nice camera. Yeah, I'm thinking wherever we see the bright white stuff might be where the most active flow is. So it's something there. Yeah. And maybe some up there, maybe some there. I bet, you see that one right in the center of the camera?
Yeah.
There's where it goes from black, there's a little bright white halo, and then there's the shrimp. So I bet somewhere in the black bit, it may be just this side. Is that boulder going to be right in your way? Just there? Let's go forward about a bio box and a half. That's the new metric of measure. People can see the bio box, you can see it in the left-right orientation at the front of the basket. It's a square thing. So, Julie, we are headed just on the top of the screen there. There seems to be some active flow. We haven't come very far. Does that look of interest to you?
Yeah, I'd like to find a 90-degree one too.
Was the last one too hot?
Too cold.
It was too cold.
I mean, it was a good one, but now that
Yep.
I know there's 90 to 100 degrees to it.
So should we try that one and see what the temperature's like?
Yeah.
There's lots more you can see on this view. There are many, many sites here, so we can take the time to dig around till we find the temperature you want.
We only have an hour and a half.
We have how long?
An hour and a half.
Well, it only takes 40 minutes to take a sample, so that gives us 10 minutes to explore. I think we're in a good place. We don't have to go more than 5 meters to do the exploration. Oh, Casey, you're such a realist. What Casey doesn't know is I adjusted the clocks last night so he had even less sleep than he knew.
Feels about right. Alright, so we're probably not going to use the mat, right?
Probably not.
Okay.
As in, definitely, if we've only got an hour and a half.
You need a sample of 40. And down.
And we won't give it an identifying of what kind of temperature sample it is until Julie tells us whether it's a good temperature or not.
Okay, so no sample.
We'll give it a sample number, but it might be a temperature or it might end up becoming a super sample instead. Depending on whether we turn the pump on or turn the pump off. So it was 50 degrees before they excavated and then
They moved
it
up
became
a
hotter
little
fluid.
bit.
Okay. You wouldn't have wanted to get another sample from the same place though.
What?
You wouldn't have wanted to get a
No.
second sample from the
No.
same place.
Not given the time constraint.
Oh, no, no, no.
So we have the super sampler in our grip, and I predict we're not going to put it back down again the rest of this dive.
And up. Alright, Alright, so where are we thinking here?
I was wondering whether the underside of the white rock just there,
Alright.
but it might be worth. I wonder if we zoomed in there, we might be able to pan left and right before we start putting the arm in and just see where else looks good when we go to the zoom flow.
Holy shrimp pension. Alright, where are we here?
So no, I think. Oh, I think if we go left a little bit, and then if we can just, if we do it in stages and then lock the camera for a bit, then it gives us an idea of it. Should be able to see where the strongest jet is. There's something just there. It looks more vigorous. Coming out just in here somewhere.
Definitely, yeah.
You agree? Might even be coming down here. These shrimp are pretty well focused, and above that, so I think maybe that little crack there. See what I mean? Yeah. Okay, that one.
Is this what you got in your room?
That's my pat on the back I got from Philip.
You got a drawing of a sailor smiling face.
Huh. I got a pair of shades and a tick. You just had all the other post-it notes in the world all screwed up and thrown all over your cabin floor. If we went back and looked at the marble now, there'd be a post-it on it saying, please pick this up. And with the joys of Photoshop, by midnight tonight, there will be.
10, 12, 14, 15,
Can you reach further away and in?
13, 12, 11, 12, 14, 15, 16, 15, 14, 13, 13,
Is that rock loose?
15,
Is it just sitting in the way?
16, 18, 20, 21,
I think the rock's sitting right in the way of the good stuff.
23, 24, 23 and a half, 24 and a half,
Yeah, it's poking there.
26, 24, 21.
Okay, so coming down this way to the right was working.
13, 12,
Okay. How about now?
15, 17, 18, 17, 19, 21, 22,
Seems like just in and around that grey boulder is where the good stuff is. We're getting up to above 20.
25.5. Still 25.5. 24.5. 23. 24.5. 23, 22, 23. 23. Very stable at 23.5.
Okay. That's not really hot enough for what you're interested in, is it, Julie?
Hotter somewhere.
Yep. So let's call that a temperature sample. Yep. So we got to 23.5. Yep. So, Julie, I recommend we move on and try and find a different place rather than
There's no hot looking place here?
at this actual. There's
No.
lots more in that field of view,
Okay, okay. in
but this was just the
particular
first one we
spot,
came
got it.
to.
Yep.
Was so there's lots of other little. We can look around with the wider field of view to see if there's something better. Yep. And we got to 23.5 was the highest, I believe. Oh, actually, you're right. 24.5. Sorry. Well done, Leighton.
I'm just gonna try to regroup this here, but
Yep.
more better. That's a lot.
So Chris is repositioning to.
You can up and left a little bit, check why they are.
So Leighton's just switching perspective, so you can actually see more of what's sitting out in front of the basket. It's not such a good quality camera, but you can see the whole hillside here is teeming and alive with fluid flow. We just stopped at the first site that looked like it had reasonably vigorous fluid flow. We checked out the temperatures there. It's about 25 degrees Celsius, so pretty nice for a hot tub, but not especially good for microbes and the limits of life. We'd rather get something closer to 100 degrees Celsius to sample. So Casey's just going to get himself a firmer grip on the temperature probe, and then we'll move forward and try somewhere different.
Alright, so where are we headed here?
Well, we'd like to pan up and use your camera to choose where to head to, if that's okay. So this stuff in the foreground we already checked out. So let's look up ahead. I'm suspecting this might be something quite good there. But if we can see anything, yeah, if we can see anything closer that's worth trying. This is where we were just looking, right? So I think the next shimmering stuff. Well, can you reach that from where we are now without moving? Casey had seen something down here. Doesn't look as...
Oh, yeah.
So it won't take long to just check out the temperature since we're already in the right place. And just got. Have we got temperatures there, Sean? Try above the rock.
About 7,
Just try and just in there more.
9,
Nope.
10. Nope, not quite 10. Dropping down to 9. 8.8.
No, I don't think that's going to get any more. Okay. Let's withdraw that. And nudge ourselves a little bit further up the hillside. I suppose it should be. But we didn't move, right? It's just a different crack.
So 14-1?
No. Sample 15, but same location and everything else, just different temperature. I think with an error we can call that 9. So it'd be interesting, that little bright white horizontal bar. If you look at that, I bet that might have more active flow than the rest of the shrimp areas.
It's all that white.
Well, I think a lot of it is alteration of rock, but sometimes when you just see like a little horizontal
Right,
lip,
right.
it's like flow coming out. So there's one just there, and there's 2 more like overhangs. I guess it must just be where the rock's just hanging in the water, but it must just be dissolved away to nothing but clays or something. Can you see any flow coming out from under that one, Casey?
There is a little, you can see some up here,
Yeah.
right there.
I was wondering if I could see something, but I don't know if I'm imagining that it's
Oh, there is some.
from that
That
one.
might be from the bottom.
Before, okay. I mean, if there isn't vigorous flow, it's not going to be interesting to Julie. I don't think. Is that true?
Right. Well, it's just
So
a
unless we
target
can,
sample.
so
What about the one?
oh, well spotted. Yeah.
Oh, so close. Zoom in.
Know exactly where it is.
7.
Zoom in.
8. 8 degrees wherever you are right now.
Roger.
About 6. 7.
I guess it may be the thing that
9. 9.5.
the whole hillside is so porous, it's hard to get high temperatures coming out of it.
Yeah.
12, 13, 16 and a half, 17 and a half, 18.2, 17, 16, 15, 13, 12, 11 right now,
What else do we need to accomplish on this stuff?
12 and a half, 13,
No,
12,
Do you have
it's
another
all you, baby.
target area?
14.
So we're going to have one last go in this outcrop. What we're finding is that the temperatures here are all sort of struggling to reach 10 or even at best 20 degrees Celsius. It seems like the whole hillside's so porous that there's a lot of seawater getting mixed in, so we have lower temperatures than we'd actually want. We'd rather be close to 100 degrees than 20 degrees. Do you want to zoom camera again? Switch camera. So we're using the Schlieren effect in the video to try and work out where we think the most vigorous flow that there might be here might exist. But I think we're realizing we probably need to move further up the hill and closer to the black smokers to see whether we can't find something a bit stronger and higher temperature. But if the shrimp like it, chances are it's just not the temperature range we're looking for.
All right, let's not bother.
No. Okay, thanks, Casey.
Shall I upload that? Pretty much like
8.3. Right, let's look up the hill again and see if we can see something that looks significantly more vigorous. How about that one just there?
Oh, yeah.
There's a couple there.
One up further.
Yep. One there and one there.
Any guesses?
Come on, baby, be a hot one.
What are we calling this area?
We haven't got that far yet. We know where it is.
Okay.
It's on the north face of the BB Vents mound. So the chimneys are directly uphill.
Okay.
So it's on that climb from Hot Chimlet to the Black Smokers.
So Hot Chimlet isn't in the BB Vents mound area?
It is. It's on the
Okay.
but it's on the north slope of the.
Okay.
So it's a big hill and the chimneys are on the top.
Oh, that's
and
right.
and the Hot Chimlet is right down the bottom, and this whole hillside, basically anywhere you go from one to the other, you go through all these other sites. So I'd kind of hoped that they were all going to be similar temperatures, but it's a bit elusive at the moment. Okay, Sean.
5.7, 6, 9.7, 15, 20,
Can we zoom?
2.
Yep. Zoom in, Ryan.
Just going to zoom the camera, Sean, so we can see.
Temperature was going up pretty fast.
Yeah. So up against that white stuff, I bet. Not just in below the white stuff.
Looks like it was made for the sampler to slide.
Slide it in and hook it.
8, 9, 10, 11 and a half, 10.2, 9,
I think it might need to go further up to the left.
7,
I think it's just off the top of this screen. You might need to pan up with the camera.
16, 21, 23,
There we go.
25,
Try to go a little left as well.
23, 22, 19,
Push in there.
21, 23, 26, 20, 18, 17, 16,
And come back left again.
15, 14,
Doing push-ups.
15, 12, 13, 14, 15, 16, 22, 23 and a half, 21 and a half, 20, 18, 11, 10.
I think we should probably get off this hill.
Okay.
Because even the most vigorous looking things, I mean, that looked promising.
Yep.
And it's deceptive.
Yep.
So, let's keep the wand in our hand. What was the best temperature we saw there? 20 something? Yeah.
So your husband's on Twitter now. He's reading or he's reading? Well, he's reading to you and I didn't notice it today. I know you're Twitter. No, he said, this is to you via Twitter. Wait, what? He said, this is to you via Twitter.
So, Julie, my best thought now is if we go up towards the top of the hill nearer to the BB vents and look for some diffuse flow sites near there.
Okay.
Is there anything that on there worth looking at? It may just be stained rock.
Flow there.
Hmm?
There's flow there. I think.
You don't, but it's hard to
Can we
get
just
about 20 here.
get across to have a look at it?
Figure out where it's coming from. So that means mixing the seawater and traveling up this space.
Actually, no, let's just keep going.
And it's hard to hold people's.
I think we're getting our eye in now, Casey. We know it's probably not worth stopping unless we see some really quite vigorous and violent flow.
Well, half a chimney is 140, so you're disgusting.
That's pretty much straight ahead, isn't it, that view there? Is that any good in there, do you think? That's probably...
A little bit of flow there.
But it's probably no better than that stuff that was 10 degrees before, though, right?
Yeah, the shrimp are rolling in it.
Yeah, so let's ignore that.
So you can imagine it comes straight out, right? And then there's another path that goes like this and mixes with seawater and hangs out and come back to the cooler and some other reactor.
There's still plenty more coming up.
Or
I'm ahead, although at some point we come into the furry walls microbes, and then we just want to fly over that.
so we don't know the fluid pathway. We don't know when or where this will occur. It could be. There's just not study enough.
Yes. Do we have that target from dive 57? Maybe if Mario can steer us towards that.
That usually zones a little bit off to the right.
And then there's the black smokers are going to be near there as well. So anywhere near there, if there's some diffuse flow in and around where the chimneys are. So this microbial stuff is uphill from that low temperature fluid area. But it seems like we did pretty well by getting the hot chimney fluid that we did get.
I know. But this was 4 days ago. They posted this.
Did we get a USBL update recently?
Well, see the USBL, those are all the blue dots.
Yeah.
So it's
And then that pink dot is us now, right?
kind of not entirely helpful.
There's a lot of anemones over there. If they're the anemones that feed on the shrimp, then that might be heading towards shrimp country, maybe? Mm-hmm. So I'm trying to get past that, because I remember there wasn't any fluid flow where the
Yep.
furry stuff was, but when you get to the anemones, you're getting close to where the shrimp areas are. And then there must surely be some good places in around where the black smokers are, right? Is there anything just coming this way? Is that same rock we're just going over? No. This much white stuff covering the rocks, there's got to be flow around it somewhere. But it may be just even weedier and wispier flow, I suppose that's the thing, it doesn't necessarily have to have the temperature to have the chemical nutrients in it. What's that coming up over on your left? Is it just more microbial map? Yep. Mm. So the chimney should be 20 meters, it's pretty much dead ahead. Infinite supply of anemones at the moment. Sure. So we're going to reset our video, so if you're watching live online, please plan to refresh your browser, and we should have everything back good to go in about 3 to 4 minutes. Is that more just anemones coming up? Yeah, it's just more of that furry microbial matter, isn't it?